Yeah, I'm recording it. So at least the picture is going to be on the screen, you know. I wanted to ask all of you, you can note one more thing. I need to change my own view. Okay. I have a question for everybody. Do you hear the sound okay? Okay, good. So again, the virulence of enterohemorrhagic E. coli and the virulence of Shigella, they all depend on the Shiga toxin. Any questions about this? Okay, so we're gonna jump to another one that didn't fit here. Another enteric microorganism, which is going to be cholera. Vibrio, I'm sorry, not cholera, Vibrio. So Vibrio species are interesting because they all prefer pH around 8. So they live in kind of a basic environment. And they survive in the, trying to see if I can, yeah, it's okay. I'm just trying to see, can you see me as the large video? I'm just trying to make sure that the recording is recording me. Look, if not, I will check the recording tonight. If it didn't record well, I'll figure something out. At least I can post the older recording of the same material. So Vibrio species, pH 8. And it also prefers salty water, brackish water. Okay. So there are 2 species in the Vibrio genus. One species is Vibrio Vulnificus. And another species is Vibrio Cholera. Vibrio vulnificus is commonly found in the ocean water in the tropical regions. It can be transmitted through water or crustaceans, and usually it leads to the wound infections. The really high mortality if it is not properly treated. Okay? We good? You following? Now, Vibrio Cholerae is another interesting gram-negative intestinal parasite. So it is transmitted through water, mostly. Also can be acquired from crustaceans. And it leads to the disease known as cholera, which is a watery diarrhea. So the volume of diarrhea is so large that people simply die of dehydration. People can have diarrheal bouts every 20 to 30 minutes. You following me so far? Now, how does the Vibrio Cholerae, how does it cause this diarrheal disease? Turns out that cholera toxin stimulates the secretion of cyclic AMP, okay, and that increased production of cyclic AMP in the epithelial cells in the intestine leads to increased intestinal secretion, which essentially leads to diarrhea. So far, we good? Wait a second. I'm playing a little bit with the... Okay, yeah. I hope it works fine. Okay. Is that clear? We're good? Any questions? I deliberately, one of the toxins that are on the study guide is cholera toxin, so I try to explain what's going on here. Okay. So we are done with the cholera. We can move on. Oh yeah, one little detail that I wanted to highlight about Vibrio Cholerae. It's transmitted not just only as an individual bacterium in the water, but it also can hitch the ride on some crustaceans, small, tiny little copypods. So if you are in the area that is known for having cholera, just fold the cloth 4 times, basically like a cotton cloth, and filter the water through that cotton cloth. And you will filter out the majority of those copypods and greatly reduce your chances to acquire this disease. Ideally, of course, you should boil the water. Questions? No, moving on. So I would say we are approaching the culmination in terms of the kind of, pathogenicity, virulence, and interestingness of bacteria. We're going to talk about microorganism from the genus Yersinia. So the genus Yersinia represents a large group of zoonotic microorganisms. I mentioned 2 of them. One is Yersinia enterocolytica, which is a typical zoonotic infection that can be acquired from semi-feral cats and dogs. So your well taken care of house pet is unlikely to carry this disease, but dogs and cats that kind of wander around can easily acquire it from the environment. So Yersinia enterocolytica causes diarrhea, gastroenteritis. Diarrhea, vomiting, it is often considered an orphan disease, meaning that you cannot, people usually don't think about it as a primary, cause of infection. Does that make sense? So an average physician will not think of Yersinia enterocolytica as the causative agent in a particular case. And, of course, Yersinia pestis, a famous causative agent of Black Plague. So the transmission route, we have to divide the transmission routes in 2. First transmission route is fleas that transmit this infection from rodents to humans. Okay. Now, Second transmission route between the humans. It's a respiratory route. Contact, if a person gets in a close contact with a corpse or someone who's infected, that's between the humans. So far, does that make sense? So, fleas essentially make this disease zoonotic. For instance, in the United States, there's a pretty large natural reservoir for plague. It's groundhogs in the states of like Nevada, California, Utah, basically Southwest. So what are the forms of this disease? So first form, that is caused by the flea bite, when flea jumps from a rodent into a human. The first form here will be bubonic plague. Okay, and bubonic plague is characterized by necrotic lesion in the lymph node, so-called bubo. Okay, does that make sense? So basically, a person gets bitten by a flea, and the adjacent lymph node where microorganisms end up becomes necrotic. It swells and forms this large black bubo. Now, respiratory transmission results in so-called pneumonic plague, infection of the lungs. If bubonic plague is only about 70% lethal, pneumonic plague is about 90% lethal. Both of these infections, both of these forms of disease can lead to so-called septicemic plague. Septicemic plague is characterized by the presence of microorganisms in the blood, and it's practically 100% lethal. Any questions so far? Okay. Good. So we're moving on to another zoonotic disease, another zoonotic infection, which is Tularemia. Francisella tularensis is the causative agent of the disease called tularemia, which is basically just a garden variety gram-negative infection. But first of all, there are two forms of tularemia. There is a cutaneous form on the skin and pneumonic form. So cutaneous form is rather mild. Pneumonic form can have a mortality rate of about 50%. So it's really, really dangerous. Now, it is a zoonotic infection. And it can be transmitted from rabbits. It's so common in rabbits that sometimes it is called a rabbit fever. Okay? So how it can be transmitted? Cutaneous form is usually transmitted when someone is skinning a rabbit. Does that make sense? Just a hunter, and the rabbit blood gets into the wound and so on and so forth. Pneumonic infection happens, it's like kind of funny. There were cases when people would just mow the lawn, and there would be a dead rabbit, and they would just plow over the dead rabbit and basically aerosolize it and inhale. They don't know, but the infectious dose is really, really low, so they would just inhale the resulting aerosol. Okay? We're good so far? Okay. So we've got just a few more bacteria to talk about. We kind of, in terms of the interest, we're going to go downhill from here. So first one here will be Pseudomonas. Pseudomonas Aeruginosa. This is essentially an environmental bacteria, but we're not concerned that much about the transmission of Pseudomonas from the environment. It is one of the most famous nosocomial pathogens. It is easily transmitted in the hospital settings and affects mostly immunocompromised patients, so we can consider it an opportunistic pathogen. The conditions associated with the infection by Pseudomonas aeruginosa, they lie in the range from skin infection and I have some really nice pictures in the lecture notes, to urinary tract infections, to pneumonia. And pneumonia is what Pseudomonas aeruginosa is most famous for. Pseudomonas pneumonia affects, to a large extent, patients with cystic fibrosis. The thick mucus in the respiratory passages provides a perfect breeding ground for that microorganism. And due to this, those patients often receive antibiotic therapy. And here stems the problem. When those patients are routinely treated with antibiotics, even prophylactically, it drives the development of antibiotic resistance in Pseudomonas aeruginosa. So it is probably one of the most notoriously antibiotic resistant microorganisms. I shortened antibiotics to AB. So far, we good? Okay. Another pretty interesting microorganisms that I'm going to put here in the center is Legionella pneumophila. You can tell from the name pneumophila that it loves to infect the lungs. So it causes atypical pneumonia. What is so atypical? Turns out that legionellosis, this pneumonia caused by gram-negative Legionella pneumophila, is a non-communicable infection. If there is a patient with legionellosis, this patient cannot transmit this infection to somebody else. Why? Because normally Legionella infects amoeba. It's the environmental pathogen of protozoa, of amoeba. It infects them and just sits there. How do humans get it? Amoeba live in the water, and water gets aerosolized for instance, from the poorly cleaned air conditioning systems, or from the cooling towers. Amoeba can be present in the water droplets. People inhale this amoeba, Legionella gets out of the amoeba and infects, in humans, it infects alveolar macrophages. But again, it is not suitable for the transmission from person to person. Does that make sense? Good? Any questions on this one? And finally, Haemophilus influenza. So Haemophilus influenza is the respiratory transmitted infection. It can cause respiratory issues like cough or sore throat or middle ear infection, but it is the most famous for causing a community acquired meningitis. Which makes it, together with Neisseria meningitidis and Streptococcus pneumonia, the third most common cause for the community-acquired meningitis. Luckily, we have a vaccine against this disease. So when you go and get vaccinated against meningitis, it's pneumococcal vaccine, meningococcal vaccine, and HIV vaccine. Sorry, I didn't. So far, we good? No questions? Okay. So we've got 4 species to chat about. Helicobacter pylori is the positive agent of gastric ulcer. Now, transmission here is kind of complicated because some people carry it around asymptomatically. You can have H. pylori and not have ulcer, okay? It's found in the microbiome. It's person-to-person transmission. It's not very clear how we acquire it. The mechanism is kind of interesting. So the enzyme called urease generates ammonia, which increases pH in the stomach locally. So it protects against acid. And the enzyme called mucinase, it breaks down mucus. Destruction of mucus exposes epithelium and leads to epithelial damage. Okay, and epithelial damage, that's your gastric ulcer. The evidence towards the fact that it is the cause of gastric ulcer is kind of overwhelming because we do know treatment with antibiotics will actually cure the disease. Okay? Give me 30 seconds. I'm going to do something to lighting. Maybe it will make it better. Okay? Okay. So we have now this was the last gram-negative. Next 3 microorganisms that I'm going to chat about are not gram-negatives. They have different cell wall structure, they considered what's called D-derms. So do not put them in the gram-negative or gram-positive category. And the first D-derm, the most boring one, I'd say, will be Leptospira interrogans. It is a zoonotic infection. That can be acquired by contact with the urine or feces from cats and dogs. Okay. The transmission, again, usually leptospirosis is more common in a farm environment because cats and dogs in the farm tend to wander around rather than just, staying in the house. The infection that it causes is leptospirosis, which in most cases in immunocompetent individuals manifests itself as a bad fever. Does that make sense? Sometimes it progresses to so-called Weil's syndrome with liver and kidney failure and splenomegaly. It's pretty bad. Weil's syndrome is bad. Yeah, I also have multiple cups stashed around me in the basement. Second D-derm. It is Borrelia burgdorferi. Okay, so Willy Burgdorfer, with a tone-breaking last name, actually died a couple of years ago. So Borrelia burgdorferi is the zoonotic infection with a really interesting life cycle. It circulates between the rodents and the deer. It is transmitted by ticks. Okay, and it causes Lyme disease. This is something kind of relevant for the state of Ohio. If you get bitten by a tick and a few days later you find so-called bullseye rash at the site of the bite, which looks like this. Okay, it's called erythema migrans, or bullseye rash, like a bullseye. You should run to the doctor and they will give you antibiotics, most likely doxycycline, and you will run a long course of antibiotics, because if you don't, Lyme disease can become chronic and it can lead to arthritis, even paralysis. So it's a pretty bad disease. Okay, and finally, the most fun of them all is, of course, Treponema pallidum. Causative agent of syphilis. There are milder forms known as Bajil or yaws. So it is obviously a sexually transmitted disease. Now the question that often arises, basically, if the person has syphilis, can this person transmit syphilis via personal contact, like shaking hands or something like this? The answer is no. Syphilis can be transmitted from mucosa to mucosa. So this is why it's STD. And we are talking about reproductive mucosa, not mouth. Okay. There were cases when syphilis was transmitted between people because they shared a towel. And one of the most bizarre cases, people shared underwear. I don't know why, but that happened. So there are 3 stages: primary syphilis, secondary syphilis, and tertiary syphilis. Primary and secondary stages are infectious. Tertiary stage, tertiary syphilis, is not. So primary stage, primary syphilis, is characterized by a lesion, a wound, on the mucosa of penis or vagina called chancre. If it is left untreated, and it is, syphilis is fairly easily treated. If it's left untreated, it develops into secondary syphilis with rash all over the body, low-grade fever, but in fact there are multiple lesions on the genitalia usually. And then if the secondary syphilis is left untreated, then it develops into tertiary syphilis when, at this point, microorganism spreads to all the organs, leading to neurological symptoms called neurosyphilis, may lead to the formation of aortic aneurysm, so it infects the walls of the aorta, or in some cases, in many cases, it actually spreads to the connective tissue, leading to lesions in the connective tissue called gummas. You will not find any pictures of tertiary syphilis in the lecture notes for one main reason: they are extremely disturbing. Okay, you can take a look. They are really, really disturbing. Okay. Now that concludes our conversation about bacteria, and we are moving on to fungi. That's going to be really short. After fungi, I will ask for a 5-minute break, and then we're going to talk about viruses. So, fungi. There are 3 main types. Molds, yeast, and dimorphic fungi. So molds are multicellular fungi. They consider to be multicellular. Some scientists are still debating whether they are truly multicellular fungi. They consist of hyphae that form mycelium. So I believe you have seen molds before, like the mold growing on the bread or on the fruits or berries. You know what I'm talking about. And it has that fuzzy appearance. So that fuzzy appearance is mycelium. And individual fibers that form that mycelium are called hyphae. Hyphae can be, so they can be septate versus non-septate. So difference between septate and non-septate hyphae is that septate hyphae, actually you can see individual cells. In non-septate hyphae, all the individual cells fuse together in one gigantic syncytium. Functionally, hyphae can be divided into reproductive versus vegetative. And the difference here, again, is kind of obvious. So reproductive hyphae, responsible for, well, reproduction, and vegetative hyphae is responsible for feeding. So far, we good? Okay, awesome. So other strategies for reproduction in molds. It can be hyphal pinching, sexual spores and asexual spores. Okay, so basically you've got three strategies. Okay, see, sexual spores, asexual spores, and via reproductive hyphae. Now, yeast, considered to be unicellular, and their reproductive strategies involve budding, and just like with molds, asexual spores and sexual spores. Now let me pause for a second and explain what I mean by sexual versus asexual spores and budding. So in case of yeast, what is budding? So budding is when a yeast cell divides, and this new daughter cell is called a bud. These yeast cells can lead to the formation of so-called pseudo-hyphae. A chain of, okay, I'm just checking that, nobody's in the chat. A chain of the yeast cells that are basically attached to each other. Does that make sense to you? Good. So chain of the yeast cells that are attached to each other. Each of them is individual microorganism. They just form a chain that scientists call pseudohyphae. Now, what's the difference between sexual and asexual spores? Well, sexual spores are formed under the harsh environmental conditions, and they contribute a little bit to the genetic diversity of fungi, which is important under the harsh environmental conditions. Asexuals, they're not really common. Sexual spores are not common, okay? Asexual spores are really common. Okay, and there are two main categories that I want you to know for asexual spores, and it is true for both yeast and molds. One of them is sporangiospores, and another is conidiospores. Let me draw the lines here, kind of signify that both yeasts and molds can form sporangia and conidiospores. So what is a sporangiospore? Imagine you have a structure. Imagine that you have a structure called a sporangia, okay? Like a big sac, okay? Inside of that sac are a bunch of spores. When the sac bursts, those spores are distributed. Does that make sense? Conidiospores, slightly different story. We have long reproductive hyphae, and conidiospores are formed at the end of the hyphae, and they just detach from the end of the hyphae and fly away. So that's the difference between sporangia and conidiospores. Any questions? Oh, yeah. Sorry. Before I ask any questions so far, I'm going to define dimorphic fungi. Dimorphic fungi, they grow as molds in the environment, and they grow as yeasts in humans. We've already discussed such microorganisms. Histoplasma capsulatum is an example of a dimorphic fungus. It grows as a mold in the forest, but when it gets into the lungs, it starts to reproduce like a yeast. The trigger that most likely switches them from being a mold to being a yeast is a temperature. So far, we're good? Any questions? Okay. So let's chat about some fungal infections. I must admit, I don't like fungi. I mean, I like eating them. I don't like them as microorganisms. So let's get it over with and move on to the best microorganisms ever, viruses. So first, I'm going to put it as a cutaneous mycosis, but a little sort of a side note, some of them are not just cutaneous, they can also be mucocutaneous. So there are three kind of broader categories. The first category, I'm going to put it as, how should I formulate it? Kind of a ringworm. So there are three genera: Trichophyton, Epidermophyton, actually there are four of them: Microsporum and Malassezia. So these four microorganisms, they all cause, I mean, okay, are you familiar with the ringworm? That's it. It's not a ring and it's not a worm. It's actually a fungal infection of the skin. I'm going to put ringworm here. And depending on the location, the disease has different names. If it's on the head, it's Tinea capitis. If it's on the that chin part, Tinea barbaris. If it's if it's on the body, it's Tinea corporis. You don't really need to. Like I have some outlined in the study guide, but those are pretty obvious, so there's not going to be anything tricky here. Second one is Candida albicans, most specifically. And this is why I was kind of so unsure how to categorize them. Candida albicans can infect nails, but it doesn't have to be only on a, like cutaneous surfaces. Candida can also infect mucosal surfaces, leading to candidiasis, which can be vaginal or oral. And oral candidiasis is, I don't remember the spelling. Is it with H or without? Thrush? I think it's with H. Yeah. So thrush is oral candidiasis, okay, fungal infection. And even more so, I was kind of hesitant to name them cutaneous mycosis due to the Aspergillus, okay? In this case, it's a secondary infection. This is what it means. You have systemic aspergillosis, which leads to cutaneous one. Let me explain what I mean by this. So imagine somebody gets infected with Aspergillus and develops a systemic disease. As the result of that systemic spread, sort of from within, the person develops cutaneous aspergillosis. Does that make sense? Quite frankly, if a patient has systemic aspergillosis, cutaneous form is the least of this patient's worries, because systemic aspergillosis is bad. Questions? Not yet? Okay. Neurotrophic infections. So there are two here that I want you to know. One is Cryptococcus neoformans. I think it's not PH, but just F. So Cryptococcus neoformans causes cryptococcal meningitis. It is a zoonotic disease transmitted to humans via inhalation of aerosols formed by bird droppings. And it's a hallmark infection of the AIDS patients. Now, one of the important virulence factors for cryptococcal meningitis is a pretty thick capsule that protects individual yeast cells, it's a yeast, from destruction by phagocytes. Now, another microorganism that can cause is Coccidioides immitis. So coccidioidomycosis also causes meningitis. It is also a zoonotic infection. Usually it can be found in the droppings of various animals, and it is transmitted to humans by inhalation of the aerosols. There are some nice commonalities that make it easier for you to study fungal infections, like virtually all of them that cause some kind of systemic disease, they are transmitted by respiratory route. It is incredibly infectious, incredibly contagious, okay? And it affects mostly immunocompromised patients, making it an opportunistic pathogen. Okay? We good? I know, I see that some of you are still writing. This is why I take a bit of a pause here. Okay, we got it. And finally, we have, so here's my plan. I'm going to talk about respiratory mycosis, then I'm going to request 5-minute bathroom break. And then, go ahead.
Sorry, what's the, so zoonotic for the
Coccidioides? Yes, they are, virtually all of them are zoonotic. Yes.
What is, like, what's the?
Not a specific reservoir. Just not a specific reservoir. Okay. So the problem with fungi is that you can't like clearly identify, oh, this is only neurotrophic, oh, this is only respiratory. So kind of only respiratory. And like big fat quotation mark. So first one that I want to highlight here is Pneumocystis juravecii. Pneumocystis juravecii is quite an interesting infection. It is an environmental pathogen. It's found in the environment, and it causes pneumonia, fungal pneumonia, with the typical signs and symptoms: shortness of breath, cough, non-productive cough, tightness in the chest, stuff like that, low-grade fever. But it is one of the hallmark disease in AIDS patients. It's one of the most common secondary infections in people with AIDS. Okay, does that make sense? Now, another microorganism worth mentioning is blastomyces. Can cause skin infection and pneumonia. And finally, the third one is Aspergillus. So Aspergillus, you know it as a black mold. Can infect lungs and lead to the formation of the hyphal balls. So think of it this way. You have seen molds, have you? Like the fuzzy growth and different structures, right? Now imagine that this fuzzy growth isn't on the bread or on the berries, but inside of your lungs. That makes sense? So we were talking a lot about different infections, fungal infections. And look, I'm not asking, you don't have to nod, just kind of acknowledge it to yourself. Some of you had ringworm when you were kids. It was totally fine, right? You got treated, you recovered, everything was okay. So cutaneous infections for adults, for immunocompetent individuals, is basically as far as the fungal infections go. Those systemic diseases, respiratory fungal infections, neurotrophic fungal infections, they all affect, first and foremost, immunocompromised individuals. Okay? That's important to remember. Fungal infections affect immunocompromised individuals. They're rare, therefore. What is interesting, why fungal infections, systemic fungal infections, are not frequent in immunocompetent ones? Most likely because of the body temperature. Humans are much warmer than the environment. So that serves as the trigger for fungi not to grow. They really don't like it being too warm. So when they grow in the environment, ambient temperature, 70 degrees Fahrenheit, they're comfortable. When they get into 98, that's bad. That's too warm. They don't really grow. I mean, they do, but very poorly, and our immune system destroys them. But in people who are immunocompromised, they can still reproduce. Okay, so give me 5 minutes. I'll come back and we'll chat about viruses. Sounds good? All right, I'm back. All right, everybody, here we go. Everybody's here? All good? Okay, okay. Well, I want to say, not that I'm going to badmouth about other people, but particular thank you for Allison and Grace for turning your videos on, because it's nice to have some kind of a back and forth. So when I say, like, does it make sense, and you're just nodding, that's good. That is really nice of you. Okay. Viruses. So I'm going to touch on some really important concepts about viruses. So what makes them so unique? And I apologize ahead of time if you hear barking. Sorry about that. Obligate intracellular parasites. Viruses cannot reproduce outside of the cell, period. Okay? Why? Because they have no ribosomes and no own metabolism. Does that make sense? They cannot make their own ATP, and they cannot run protein synthesis by themselves. That's it. First virus discovered was a plant virus, So we can say plant viruses first, bacterial, I think bacterial and animal viruses second were discovered afterwards. I don't remember the order. I think bacteriophages were second and animal viruses were third, but plant viruses were discovered first by the guy named Dmitry Ivanovsky. So he studied tobacco mosaic virus. So he mushed the tobacco plant, filtered all the crap out, had this kind of, lysate of the cells, and then he ran it through the filter that removed all the bacteria. And when he removed all the bacteria from that lysate, turns out whatever went through the filter was still infections. And he called that virus, the tobacco mosaic virus that he discovered, a filterable agent. Okay? Now, viruses can be divided into two groups. Naked viruses and enveloped viruses. So naked viruses consist of a genome inside of a capsid. Okay. Enveloped viruses consist of a genome inside of a capsid, which is further surrounded by the envelope. So we can say, the purple is genome, the green is the capsid, and this brown one is the envelope. Capsid is formed of proteins. And the envelope is proteins and phospholipids. Essentially, virus, when forms envelope, it steals the part of the cellular membrane. It can be extracellular, like cytoplasmic membrane. It can be, what should I call it? Golgi complex membrane. It can be endoplasmic reticulum membrane, all kinds of membranes. So far, we good? So those are structural features. Got it? Now, let's address a couple of questions. I'm going to remove this blue part here. Or leave it. The types of the viral genomes. So viral genome can be DNA molecule or RNA molecule. Okay? So DNA can be single-stranded, like in the, single-stranded DNA, I'm sorry, my bad. So single-stranded DNA, like in parvoviruses, or double-stranded DNA, like in herpes viruses. RNA can be double-stranded RNA, like in the rotaviruses, or it can be single-stranded RNA, like in yellow fever or HIV. And moreover, single-stranded RNA can be positive and negative. So what is the difference between positive single-stranded RNA and negative single-stranded RNA? Now, you may remember the dogma, central dogma of molecular biology, that DNA, double-stranded DNA, is used for synthesis of a single-stranded RNA that is used as a template for translation, for synthesis of proteins. So essentially, positive single-stranded RNA can be immediately translated. So for instance, you can take the RNA molecule, hypothetically, you can take RNA molecule from yellow fever virus or poliovirus, and you can use it as a template to make viral proteins. Now, if you take negative single-stranded RNA, like RNA from Ebola virus, you can't use it for protein synthesis. You have to convert it to positive RNA first. And how you do it? It's a complementarity rule. So basically you have a single-stranded molecule, and based on that negative single-stranded RNA, you make a complementary copy, which will be positive, that you can use for translation. It's like, you know, in the photograph, in the old photographic craft, you have a negative and you have a positive. Negative is on the film, positive is on the photo paper. So far, we good? Any questions? All right, now let's chat a little bit about the structure of the capsids, types of infection, and briefly overview the rest of the viral diseases. Okay, so capsids. Three main types: helical capsids. Sometimes they're called spiral, when individual proteins form a helix. Icosahedral capsids. Dude. Well, try to think of a soccer ball, okay? With all those individual patches, like pentamers that are attached to each other. So icosahedral capsid has 20 sides. It's the most common type of capsid among the viruses. And then there are complex capsids. They don't fit into any category. For instance, the capsid of a rabies virus or the capsid of the pox virus, they don't really fit into any category, so this is why we call them complex. Example of a complex capsid is the T-even bacteriophage. Okay, which has its genome packaged here, okay, in this part, that has a tube, okay, and has tail fibers and the tail pins. So tail fibers, and it actually looks like this. It looks like a little alien spaceship, okay? And these tail fibers are used to attach to the cell, and tail pin is a really interesting structure. It burrows the hole in the cellular membrane and injects the nucleic acid into the cell. Does that make sense? So that serves as a really nice segue to the process of bacteriophage infection. First of all, bacteriophage. Which color should I use? bacteriophage infects only bacteria and archaea. It does not infect mammals, insects, fish, doesn't matter, plants, no. Bacteria and archaea. Did I make myself clear? So here's how it happens. So first, we're going to talk about lytic infection. And I understand that you don't have that convenience. I can erase things. You cannot because it's paper. But it's just much easier to draw it step by step like this. So this is your garden variety bacterial cell. Where did I put another? Okay, there we go. And this is your bacteriophage. Okay. That's bacteriophage DNA. So first step would be, look, first step is attachment. Does that make sense? Once the bacteriophage attached to the cell, it injects its DNA into the cell, and we call this step penetration. Okay? Inside of the cell, this viral DNA, the viral genes, bacteriophage genes, get expressed. So that produces viral proteins, and it produces more copies of viral DNA. That process is called biosynthesis. Finally, viral proteins and viral DNA forms the get together and form a virus particle. This is called four-step assembly. And this viral particle plows through the cellular membrane and this one is, now I'm going to use just one term, egress, but it's egress, release, doesn't matter. Does that make sense? Three things that I want to highlight. First, capsid. You see capsid here? Capsid never gets into the cell. Only the genome does. Third, the genome of the virus does not get incorporated into bacterial genome. Third, bacterial cell almost immediately dies. I can tell you how much time it can take for a bacteriophage like this to kill a bacterial cell. 30 minutes. From the time it binds to the time it gets out and bacterial cell is exploding, it's 30 minutes. Does that make sense so far? We good? All right. So that was lytic infection. Infection that leads to the lysis, to the killing of the bacterial cell. Another type of bacteriophage infection has slightly more steps, and this type is called lysogenic. Now this time I'm not going to name particular steps, but I'm going to explain them in the details. So we again start with the bacterial cell, and there is bacterial DNA here, nucleoid, right? Well, moreover, hold on. Remember, bacterial chromosome is circular. Okay? This is your phage. With the phage DNA. So first step is standard. DNA gets into the cell. Does that make sense? Look what happens next. Bacterial DNA gets incorporated into a viral DNA. Does that make sense to you? Sorry, but the other way around. Viral DNA gets incorporated into the cellular, into the bacterial DNA. This is called a prophage. Okay, we're good? At this point, nothing happens to the cell. It doesn't die. And it can live like that for multiple generations. And what's cool about it? Every time this bacterial cell divides, prophage also divides. So virus effortlessly spreads through the population of bacteria. Does that make sense? Every time there's a daughter cell, it already contains a prophage. Now, what if the environmental conditions become unwelcoming? Then the prophage gets excised viral genes, so I'm going to put gene expression, okay? Gene expression from that excised prophage leads to the assembly of the mature virions, and these virions get out, essentially killing the cell. Look at the difference between lytic replication and lysogenic replication. In the lytic replication, virus does not incorporate its nucleic acid into the bacterial DNA. The cell dies almost immediately. In lysogenic replication, virus does incorporate its DNA into the bacterial chromosome. Cell stays alive for some time and does not die immediately. Does that make sense? Am I clear? Are we good? Okay, cool? Awesome. So both lytic and lysogenic types of replication contribute to the gene exchange among bacteria because these microorganisms, the bacteriophages, can carry fragments of bacterial DNA from one cell to another. And the mechanism for the gene exchange between bacteria that's mediated by bacteriophages is called transduction. Okay? So far, any questions? Okay, let's move on to the infection of eukaryotic cell. So basically an infection of a typical animal virus. And I want to, make sure that I'm clear. Viral world is extremely variable. It's extremely diverse. So there are many aspects of the infection that we simply cannot cover in this course, but I'm going to make kind of a general idea. So this is the eukaryotic cell. This is the nucleus. Okay. So virus here Now, look, I'm going to be using non-enveloped virus, a naked virus, but the mechanism is pretty similar for enveloped and non-enveloped viruses. So first, virus is to bind to the receptor. Okay, so that's first step called attachment. Okay, that's to bind to the receptor. Good? Now look, there's a first difference between the bacteriophages and animal viruses. Virus gets taken into the cell, in this case, for instance, via endocytosis. So you see, not just the nucleic acid gets in, the whole capsid gets in. Does that make sense? The whole capsid gets into the cell. So this step is penetration. What happens next is really interesting. So endosome, this endosome, usually acidifies and it breaks down the capsid. So you've got nucleic acid and capsid is gone. Those are fragments of the capsid here. Okay. So this step, when capsid is gone, is called uncoating. So far, does that make sense to you? Now, what's going to be next? Well, the question is whether it is an RNA or DNA virus. If the genome is RNA, stays in the cytoplasm. Now, DNA viruses go to the nucleus. Let me expand a little bit on this part. First, why RNA viruses stay in the cytoplasm? Presence of RNA in the cytoplasm is totally normal. Does that make sense? Presence of DNA in the cytoplasm is not. So for the virus, first of all, it's kind of a good hiding place. I'm in a basement. The entire house is empty, and I have no idea what's going up there. For the virus, it kind of makes sense to hide in the nucleus where DNA is normal. Okay? And another reason for it, look, DNA is in the nucleus, and the machinery, cellular enzymes that replicate DNA are also found in the nucleus. So virus may just use this cellular machinery to reproduce, to replicate its DNA. Does that make sense to you? Okay. Next step, step number 4 here is biosynthesis, which results in the generation of multiple copies of viral genome and viral proteins. And then they come together during the step called assembly. Sorry. Okay, so that's assembly. And then assembled particles get out of the cell. So this is release or egress. Oh, thank you. Oh, I thought my head would explode. All right. Let me go over it one more time and mention a few things. So first of all, virus attaches to the cell. Virus can attach to the cell only if there is a receptor. if the cell has a receptor, it is called susceptible cell. If the cell has a receptor, it is a susceptible cell. Virus gets into the cell, for instance, by endocytosis, undergoes uncoating. If it's a DNA virus, now, when I say it's a DNA virus, it goes in the nucleus. There are exceptions. Pox virus, which is a DNA virus, like smallpox, it does not go in the nucleus, it stays in the cytoplasm. Influenza virus, which is RNA virus, goes in the nucleus. So there are some exceptions, but the general rule, DNA viruses replicate in the nucleus, RNA viruses replicate in the cytoplasm. And reasons I mentioned before. Now, biosynthesis. If the cell can support biosynthesis, it is called permissive. Okay? So in order for the virus to successfully infect the cell and replicate in it, cell has to be both susceptible and permissive. And then, you know, synthesis assembly. Now, when viruses are released from the cell, if it's a naked virus, the release will most likely result in a cell lysis and death. If it's an enveloped virus, cell can survive for some time because virus is basically just popping through the membrane, taking the part of the membrane with them, and the membrane fuses behind it. So naked viruses usually lead to the cell death. Enveloped viruses, cell can survive for a bit. Yes, yes. So, look, Grace. Susceptible means that virus can attach to the cell. Permissive means that cell can permit viral biosynthesis. My favorite comparison, like, you know, if you're running away from the zombies and you have 3 rooms in front of you, if you have a key to one room and you open it, but it's absolutely empty, bless you, you can get into that room, but you're going to starve there to death because there's nothing in. Second, that's susceptible cell, but not permissive. Second room, you don't have a key, but it has plenty of food. So cell is not susceptible, but theoretically, it is permissive. And finally, a third room, you have a key to it, susceptible, and there's plenty of food in it, so you can survive zombie apocalypse there. So it's both susceptible and permissive. Does that make sense? Good? Okay. Let's have some fun with viruses. Okay. We're going to start with what I think we're going to do. Here's my plan. Kind of, you know, digression here. I think I recorded a really good lecture on the viruses. It's really interesting and so on. I strongly encourage you to kind of listen to it. I'm going to touch on some features of the viruses, okay, kind of highlight, and we're going to wrap up after the viruses. Next week, on Wednesday, you're going to take an exam. I'm still thinking how to do it. Most likely, listen, there's going to be a friend of mine who's also teaching at Lakeland. So we're going to do the WebEx, okay? And since I'm going to be out of town, he's going to be proctoring you. So he's going to watch you, okay? I will turn off my camera. I will kind of walk around with the phone. Will not watch you. If you have a question, you can email me. You can shoot a message in the WebEx, but you should be fine, okay? Does that make sense so far?
I have a question on our exams.
Right, okay.
So next week will be exam 4.
4.
And then we have 5 and 6 left?
Yes. And that's
Okay.
going to be Wednesday and Wednesday.
Okay.
And there, remember, exams are not comprehensive. So your exam number 5 will be on bacteria and fungi. Exam number 6 will be on viruses
Okay.
and protozoa. Does that make sense?
Yeah.
I'm going to be back for the exams 5 and 6. And exam 5, which is going to be on December 2nd, we're going to do a review of protozoa and helminths after the exam. Okay, we're good? All right. So let's chat. Okay, first one, influenza. It is, isn't it? So, things that I want you to, hang on a second. Okay. Influenza. RNA virus, single-stranded, negative. So single-stranded RNA negative. Interestingly enough, it is segmented, and that will be important in the future. Enveloped. There are 2 key proteins in the envelope. Those are hemagglutinin, H, and neuraminidase. N. So here's the thing. Hemagglutinin binds to the receptor. So it's responsible for virus attachment and penetration. Neuraminidase is an enzyme that is critical for both penetration and the release of the virus. Am I clear? You all are pretty familiar with the, how we call, influenza viruses, something like H1N1, right? You're familiar, H3 and 2. So those are different types of hemagglutinin and neuraminidase. Does that make sense? There are certain combinations that can infect humans. So this disease is zoonotic. Now you can ask, hold on a second, it can be transmitted between humans. Yes, it can, absolutely, yes. But new strands of influenza rise using the following path. So it goes waterfowl, like ducks and geese. In waterfowl, the infection is intestinal. They transmit the disease to pigs. And they transmit disease to humans. Does that make sense? Am I clear? This is, remember in 2009, the outbreak of the swine flu? That was exactly what happens. What happened? Waterfowl virus got in the pigs, from pigs got to humans, okay? Now, can we acquire influenza directly from the birds? Yes, we will chat about it like in a few minutes. Well, actually, let's talk about it now. So, I apologize, I need to look at my kind of cheat sheet here because I can never remember which receptor is where, and I'm going to explain it. So, 8, 2, 3, it's in the birds. Okay. So, in the birds, look, in the birds, infection is in the intestine, right? So, bird GI tract, the receptor for avian flu, the bird flu, like H5N1, the receptor is alpha-2,3 sialic acid. Please do not memorize this shit, like the numbers and the terms. Please do not. Human upper respiratory tract, like pharynx. We have alpha-2,6 sialic acid. Now, look, those are two different receptors. Does that make sense? So H1N1 can bind to this one, and we can have a flu. H5N1, which is a bird flu, cannot bind to this receptor. Are you following me? I'm going to address Ellison and Grace because I can see are following me. Bird flu cannot bind to this receptor. Does that make sense? So we cannot have an infection in the upper respiratory tract. But in the human lower respiratory tract, we have both alpha-2,6 and alpha-2,3. So if you inhale H5N1 really, really deep, like in your bronchi, you're going to have this bird flu infection of your bronchi, which is going to be really severe. Does that make sense? But you cannot transmit it to another person because okay, you coughing, fine. You coughing the bird flu out. But another person will inhale it and the bird flu will not bind to the receptors in the upper respiratory tract. That makes sense? Essentially, this is my favorite analogy. Can you hypothetically transmit bird flu from person to person? Hypothetically, yes. So the sick person has to cough directly into the mouth of a recipient while the recipient takes a really, really deep breath, which doesn't happen, obviously. That makes sense? Okay. We're good about that? Now, I'm not going to talk about symptoms of the flu. We've all been there, right? What I want to say, first, treatments. They are not very effective. The standard treatment Tamiflu is effective if it is prescribed early, which often does not happen. And now we have a new one called Xofluza. I don't know how well it works, really. No idea.
I've taken the Tamiflu before, and it gave me like worse symptoms than I had.
It probably didn't. It just didn't work. So, for two reasons. Imagine, so I can give you numbers, Grace. So, if you take Tamiflu, if my memory serves me well, 24 hours after the first symptoms of infection, you shorten the length of your disease by a day. If you take it in the 24 to 48 hours, so like 2 days after you started feel sick, you shorten your symptoms by a half day. So it's not particularly fantastic, you know?
Mm-hmm.
Ideally, like you get infected and you take Tamiflu right away, which doesn't happen. In your case, you may have taken it like really, really late and just didn't do anything.
Yeah. It made me really sick to my stomach and like
yes,
I was throwing up.
That is a very well-known side effect of Tamiflu. It does make people throw up and, you know, have all kind of gastrointestinal upset. Yeah, that's known. So treatments are not really. I'll explain why. What about vaccine? We all know vaccine. By the way, please get your flu shot. I know some of you have to because you work in the healthcare. Those who don't have to definitely should to. And if anyone is kind of in doubt, Google the website called Families Fighting Flu. It's the personal stories of people who lost their loved ones to influenza. It is really, really sad. So go get your flu shot. I did that they didn't even take me into the doctor's office. They put me into some shady room, stepped some syringe into my shoulder, and that was it. I don't even know if it was a flu vaccine. Just joking. Families fighting flu. And they have personal testimonials. It is gut-wrenching to read. Because if you think that, oh, flu kills like really old people, half of those testimonials are about people your age, like dying from flu. It is, so definitely, vaccine should be taken. Vaccine sucks. It's not good. I mean, it's safe, but it's not super efficient. But it reduces the symptoms, even if you catch one. It makes you not as sick, and it apparently reduces transmission. It definitely reduces mortality. Kenny, you have a question?
No, I was just gonna tell a story. I actually lost a friend 4 years ago to the flu. She got really, really sick, and her parents wouldn't take her to the doctor and give her the vaccine, and she ended up dying from it.
That's sad. That's really yeah. Sorry to hear that. That's terrible. The worst part of it is that it's totally vaccine preventable.
Yeah, I don't know why. She was already sick. She just had a minor cold at first, and then it got 10 times worse, and in 3 days she was gone.
It's it is. I mean, I can kind of understand with the coronavirus what's going on now. I mean, we do have treatments, but with the flu, just have to get a vaccine. And all health insurances cover the cost, okay? And even if they don't, it's totally worth, I don't know, what, $25 or $50 that it costs. And just, yeah, it's gotta be done.
Can you explain like the new coronavirus, why can't I think of what I was going to say? They're like gonna take RNA
for the vaccine?
Yeah.
Oh yeah. Let me finish the flu vaccine story, and I'll do it. It's like super easy. So, flu vaccine, virus changes, and that whole conversation about vaccine was a preface to the story that virus changes. And this is how it happens. So you see the segmented, that it's segmented. So first of all, and it's RNA. So when RNA replicates, it accumulates mutations. Because RNA synthesis is very, very imprecise. Does that make sense? By nature. And that's remind me about RNA synthesis when I'm going to mention coronavirus. It's very imprecise. This slow accumulation of mutations, and I want to make it clear, it is slow. This is called antigenic drift. So our garden variety H1N1, this here is different a little bit from H1N1 from last year. Does that make sense? Because it replicates when RNA is copied, it accumulates mutations, and it slowly drifts antigenically away from the original. Now, segmented. I don't have a prop here, but you can easily imagine that. So let's say I have 8 cards, like cards, okay, playing cards. You know what I'm talking about. And Allison has 8 different cards of the same type, which is going to be spades. So hearts, and Allison has spades, okay? Now, she has 8, I have 8. So we put those stacks together, we shuffle them, and we deal 8 cards to each other. She still has 8, and I have 8. But now she has a mixture of hearts and spades, and I have a mixture of hearts and spades. Does that make sense? This random reassortment of genomic segments is called, I'm going to put reassortment of genomic segments. It's called antigenic shift. So you see a terminological difference, the drift and the shift. Drift is slow, shift is rapid and abrupt. Another term, like next term, look, it's not going to be an exam. I just really like the term. It's the concept related to the flu virus. It's called original antigenic sin, which implies that when you were, say, you were 3 years old and you got your first flu infection, does that make sense? That flu infection, the first one, shaped your immune response for life. And you are not going to respond really well. You're not going to form a robust immunity to any other flu infection except your original one. I mean, you're going to form some, but it's not going to be too good. And that is often considered to be a problem in the flu vaccine. Okay, we got this racist coronavirus question. How does RNA vaccine work? Okay, that's exciting. So, this is your viral RNA. So basically what they do, here's what they do. Look, they take a part of the coronavirus RNA genome. Coronavirus is the RNA virus. They take a part of coronavirus RNA genome and they artificially make that specific part of RNA genome that encodes coronavirus glycoprotein. You probably have heard multiple times. It's called spike protein. Okay. Spike protein gene. Obviously, it's not a naked RNA. It is packaged into a lipid coating. It's all like technology and stuff. That RNA gets injected, and that RNA vaccine encounters the dendritic cell. Okay? Does that make sense? Remember dendritic cells, antigen-presenting cells? So dendritic cell takes the RNA in. And inside of the dendritic cell, now this is some hardcore molecular biology, and I'm not really sure how they did that, but somehow they did so that this RNA serves as the template for spike protein synthesis. Okay? Inside of the dendritic cell. And look what happens here. Dendritic cell, being antigen-presenting cell, presents spike protein. More strictly speaking, it's like some fragments of spike protein, but we're going to simplify it a little bit. It presents spike protein to your adaptive immunity. So presentation by MHC class 2 will trigger CD4 response, the T helper, and it will trigger B cell response, antibodies. Does that make sense? Presentation by MHC class 1 will trigger CD8 response. So cytotoxic T lymphocytes, the killer response. So what we know is that this vaccine, the RNA vaccine, makes a really good B cell response, a really good, robust antibody response. And from what we know so far, remember, when did vaccine, so phase 1 trials were started in March, in freaking March. It is November. It is what, 8 months? It's insane. In 8 months, Moderna completed phase 1, phase 2, and they are in the phase 3 now, and they have data on the levels of antibodies, and they can compare those levels to antibody levels in people who recovered from the disease. Turns out vaccine gives same level of antibodies. Does that make sense? So now, T cell responses are much harder to measure, okay? Because ideally, what we want to take a fragment of a spleen or a fragment of a bone marrow or a fragment of a lymph node. And I don't think that participants in the study will agree on the lymph node biopsy. But some sort of methods allow us, so we can make a conclusion that cellular responses are also pretty good. So I am like, if you want my opinion, my personal one, like from my virology background, if someone will knock on my door today and tell me, dude, we can give you Moderna vaccine, right away, I'm ready. I have no doubt. It works, it's really effective, preliminary, should be fine.
Now, is this like going to be a vaccine that's going to be yearly or no, because it's going to reproduce? So
a couple of things here to kind of unravel. First of all, it looks like immunity to coronaviruses in general doesn't last long. Does that make sense? It just, even natural infection does not give you a lifelong immunity. We can be reinfected. Make sense? So most likely the vaccine will not give you the immunity against infection. It will give you the immunity against disease. Do you see the difference? So you will not get sick. You may still carry the virus around. And that's where the caveat is. Because, Grace, are you working in the healthcare? Are you working with sick patients? Now, you, as a healthcare worker, will probably be among the first people who will get this vaccine. It's actually a plan in the state of Ohio to give, essential healthcare workers first. Now, imagine you get this vaccine, you're protected. You're not gonna succumb to the disease. You're not gonna get sick, but you still can get infected and not know this. And imagine you are infected, you have no symptoms, and you walk to some 80-year-old dude with ischemic heart disease, and you give this virus to a dude, and the dude dies. That's kind of sad. So my point is, masks, we're going to wear them for a while. Okay? And, like, seriously, my main problem are stupid people. When people will not accept vaccines, say, no, I'm not going to get vaccinated, that's going to be, the more people we cover, the less the spread is going to be. That's it. Like, we need to cover, ideally, everybody. Now, will we have to take this vaccine again? Possibly, to boost the immunity. Most likely, this virus eventually will become just a seasonal cold. Does that make sense? Now, if you are asking, is this virus gonna change? The interesting feature of coronaviruses is that they do not change much. They mutate very slowly, which is unusual for RNA viruses. They have special mechanism that makes them very, very genetically stable. Okay? Did I answer your questions? Now, first of all, I love the questions about viruses, so please ask. But I'm going to post the link to the presentation, knowledge exchange presentation that we had recently. And it is a YouTube video, obviously. It's not a great sort of, like, I'm not standing in front of the board, and it's a recording of a WebEx. But it's a pretty decent recording, okay? And it may answer a lot of your questions, okay? It's, I mean, not as exciting as many other YouTube videos, but okay. So we're going to move on to, yeah, go ahead.
I was going to ask if you were going to post that because I signed up for it and I just wasn't able to come to it. So it's
yeah.
on your YouTube channel?
Oh, it's already on YouTube. I'll just post the link.
Okay.
It's on Lakeland's YouTube channel.
Okay.
Yeah. Okay, so few more viruses. These are my favorites. Flabby viruses. Flabby means yellow. They named Flabby viruses because of the yellow fever. So these are enveloped, positive single-strand RNA genome, non-segmented. So I'm going to break them down into two main groups. Well, three actually. First will be encephalitis. Second will be hemorrhagic fevers. And third will be just fevers. Okay, so encephalitis, West Nile, tick-borne encephalitis, Japanese encephalitis. Hemorrhagic fevers, yellow fever, and dengue. And just fevers, Zika. Now, let me be absolutely crystal clear. This 6 viruses doesn't even come close to cover all spectrum of flavivirus diseases. Does that make sense? There are many, many more. Transmission routes, all of them are vector-borne. All of them are transmitted by vectors. Mosquito, 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 and this poor bastard is transmitted by tick. Tick-borne, okay? Does that make sense? I'm going to do some kind of color coding here. So vaccines. Yellow fever, tick-borne, Japanese encephalitis. Okay? For West Nile, we have a vaccine for horses, but not for humans. Okay? Now, one more color coding. Transmission between humans. Yellow fever, dengue, for sure. Now, I believe Zika as well. Okay? Yeah, for sure. So yellow fever, dengue, Zika can be transmitted between humans. West Nile, tick-borne, Japanese encephalitis are all purely zoonotic, from animals to humans. That makes sense. Now, Briefly, the transmission cycle for these guys, for all of them, there is a natural reservoir, which is monkeys. So the mosquito can transmit from person to person or from monkey to a person. Does that make sense to you? Now, common course of infection for all of these, and these are pretty bad diseases. You can die from them, okay? It's a biphasic disease. Okay, so first phase, flu-like symptoms. Second phase, either encephalitis or hemorrhagic fever. Does that make sense? So flu-like symptoms, it's not flu. Like if you think that how people describe, how you feel when you have flu, you've been hit by a truck. In this case, you've been hit by a freight train and a freight train ran over you several times. It's really bad, like super bad. Myalgias, arthralgias, headaches, really high fever. But let me explain another thing. In most cases, in all those infections, after the first phase, nothing happens. You recover and you're fine. But for some percentage of folks, second phase coming, and they can die from it. Does that make sense? Specifically for Zika, there is no first and second phase, it's just a flu-like symptoms. But it can be transmitted vertically from a mother to a fetus, and it leads to microcephaly, basically a fetal abnormality. Does that make sense? No two phases, but one phase and intrafetal infection. That's not intraplacental. I don't remember the proper name, but you get this. A lot of teratogenic effects. Make sense? We're good? Now, shameless plug. I actually have a video that I can post too, like in the old times when people didn't wear masks and we were alive, about Zika virus, when it was a hit in 2017, I think. Okay, what we have next? Zika, polio. So we've got polio, Hep C, and noro. Okay, so polio, positive single-strand RNA genome, naked virus. Okay, causes, well, polio. It's the paralysis. Flaccid paralysis. It infects the neurons in the ventral horns of the spinal cord. So these neurons in the ventral horns are motor neurons. When they are destroyed, you can't move. Okay? You have flaccid paralysis. Question? Anyone? No, good. Basically, you cannot control your muscles. Now, virus transmission is fecal oral. It can be found in the sewage. It is exceptionally stable in the environment. It can be found in the sewage easily for a year. Okay? That makes sense? So paralysis infects neurons, fecal oral transmission. I'm trying to think. Yeah, people per one person. So usually it's only one case of paralytic poliovirus out of 200 infected. Does that make sense? So infection of the nervous system is accidental. Virus reproduces in the gut, okay? And generally, polio infection leads to very kind of, generalized symptoms like fever. I'm waiting for a question. Spinal cord. So there are two vaccines, inactivated polio vaccine and oral polio vaccine, okay? So inactivated polio vaccine, well, inactivated, developed by Jonas Salk, and oral polio vaccine is attenuated. So oral polio vaccine contains actual poliovirus, but those strains are weak. Does that make sense? And they cannot cause the infection except in 1 out of million people. So in 1 out of on average million people vaccinated by OPV, there can be one case of vaccine-associated poliomyelitis. So that's Albert Sabin. Now look, here's an interesting thing. Since I saw you, I have an idea how old you are. You all have been vaccinated with an activated vaccine in the United States. I was born in Soviet Russia. I was vaccinated with attenuated vaccine by Albert Sabin. And it's oral vaccine, so all you need to do, it was really cool, you had to stick your tongue out and a nurse would give you a little drop of the vaccine and you swallow it and that's it. It's really easy to administer, so it's really effective, gives you really robust immunity, but side effect, 1 out of million people will develop poliomyelitis. Does that make sense? Now, I'm going to touch on Hep C and Noro, and we're going to wrap it up. So hepatitis C, positive single-stranded RNA, enveloped virus. Few things. It infects liver. First, it's a human virus. Humans are hosts and humans are the reservoir. There is no, it's only human-to-human transmission, period. Infects liver. Chronic infection. Okay. So the distribution is approximately like this. Imagine this: quarter of the people will develop, like they will clear the virus. They will just be virus free. You will get an infection and you will be virus free. 3/4 will develop chronic infection, and the outcome can be just a liver damage, kind of moderate damage, or cirrhosis, or liver cancer. Okay, does that make sense? And it's basically like 25% will develop just a generalized liver damage, 25% cirrhosis, 25% liver cancer. Okay, we're good? What's the problem with that virus? No animal model, and it's hard to grow in cells. It is very inconvenient to work with, okay? There is really, like the only animal that you can give it to, basically humans, I believe also monkeys, but working with monkeys is really hard and expensive. Okay, mice don't work like all that. Rabbits, they don't work. Hard to grow in the cell culture, which makes it even more inconvenient to work with. So we have no vaccine. But really effective treatments. Now, I talk about it extensively in the lecture. In the notes, I also provide the price of those treatments. So to give an idea, it's a 12-week course of a drug. And the first drug on the list, I don't remember the name of it. It's Sovaldi and Gilead, Cefazbuvir. The overall course is 84 grand. So for 12 weeks, basically means that every day you have to spend $1,000 on the drug, which is not. Maybe it went down, I don't know, maybe now it's cheaper, but still, it's pretty expensive, okay? Does that make sense? Hypothetically, if we can treat each and every person in the world, since it's purely human to human, we can get rid of it. Oh, transmission routes. I don't know how highlight it more. First and foremost, it's a transmission through blood. Okay? Old sources say sexual transmission. Hepatitis C is really poorly transmitted by sexual routes. Does that make sense? There was an experiment, an actual study, clinical study that showed, no, not really. It can be transmitted, but it's really ineffective. Okay? And finally, norovirus. Positive single-stranded RNA. It's naked virus. So just as any other naked virus, it survives really, really well outside of the human body. So norovirus can cause gastroenteritis. It is the most common cause of gastroenteritis in the United States. We're talking about thousands of hospitalizations and millions of cases every year. It's also called winter vomiting bug and two-bucket disease because people have diarrhea and vomiting. It runs for about 2 days with mild fever. People recover. Very few people die of it, mostly very old people and newborns because of dehydration. For an adult, actually, I can tell you that norovirus studies on humans are allowed. You can study norovirus in people. You can give people an infection, they will get sick, and 2 days later, they will recover. That's how they used to study. Now there is the mouse norovirus was discovered, so it can be studied in mice. Transmission route, well, fecal oral. Main sources, food and water. It is extremely contagious, infectious, so ID50 is really low. So it's very easy to give it. And what's important, virus can be transmitted during incubation, disease, and convalescent stage. Okay, so basically, let me explain. If I am already infected, but I don't know it, and I visit somebody's house, they will be infected too. I'm in incubation. If I am sick, obviously, I can give it to other people. But even after I recover, I can still shed the virus for up to 4 weeks. Okay? That make sense? And for some reason, it does not confer extended immunity. So much so that you can be infected with the same virus 6 months after you recover from the infection. For some reason, we do not form a robust immunological memory against the norovirus. Okay? You can be infected with a different strain as close as 2 months after the infection, which kind of, sucks. I'm trying to think what else. We talked about cases. We talked about the transmission of the virus. Yeah, that's kind of it for today. So next Wednesday, we will figure out the proctoring part. But that's it. I'm going to post if recording is good, I'm going to post it.